Hello everyone, I'm Ziwei Yang from KyotoU University, and this is my first time to attend this CIKM. I'm really glad to be here, and I hope you can enjoy my presentation today. So, our work is titled the MoCLIM towards accurate cancer subtyping via multi-omics contrast learning with Omics-Inference modeling. This is a cooperative work from KyotoU University and the OsakaU University. So I will begin with a summary of our work and then pass to more details. So in this work, we propose the MoCLIM. It is a framework for accurate cancer subtype identification through multi-omics data. So our work composed of majorly two modules and combined with important observations from the biological studies. The first module is we try to independently learn the omics-specific features by modeling the data distributions for each omics data source. And in the second module, we try to unify these omics-specific features by the multi-omics contrast learning. And also in these modules, we put our framework in an important biological axiom, that is, the genome-wide transcriptomics is the focal point of all the omics studies. And let's talk about the background, the cancer subtyping. We all know about cancer, but actually, one cancer may consist of different cancer subtypes. And for each subtypes, they are under control of the unique biomedical control mechanisms, such as the changes in the hormone modalities will actually lead into the different cancer subtypes. And of course, each subtypes will require a different therapy strategies. However, the task of the identifying cancer subtype is not easy. Actually, in our clinical setting, our doctors always found that the different subtypes, they share the same characteristics in the pathological image or other medical image like the CT scans or something. So there are no differences in these image layers. However, actually the differences we can say that hiding behind these images. So we can find that in these figures, it shows these three different cancer subtypes. The differences can appear in the different omics layers. So in these papers, we want to use the omics data for the cancer subtyping task. To do this, we need to answer some important questions. The first one is, how can we learn a good representation from each specific omics level while avoiding some data issues? So for the cancer omics data, the omics data are usually have small sample size, and for each samples, they have high feature dimensions. So, like we can see in these figures, they have the DNA layers, the microRNA layers, and maybe the protein layers, and each of them consists of information from the different information for a sample. And the next question is, how can we effectively integrate this multi-omics data while being guided by the biological observations in the omics inferences? Like here, we may have extract the diverse individual omics features from the different omics layers, and now what we want to do is to unify them into a common feature space. So actually, in this common feature space, they will contain all the information we need to perform the cancer subtyping task. And in this feature space, there should be some cross-omics inferences relationships just between all these omics layers. And in most cases, we should consider the transcriptomics as the focal point. Like shown here, at the beginnings, we may have DNA sequences, and after the transcriptions, we get its messenger mRNA transcripts. And next, these transcripts may be translated into certain proteins, which may have the functions to regulate the downstream microRNA transcript. So we can find that all these layers, the DNA layers or the protein layers, they are actually related with what we think the most important is the mRNA transcript layer. So to answer these questions, we posed our framework. So this framework consists of 3 important modules. In the first one, we use the unsupervised generative models to independently learn the data distributions for each omics layers. And for the next, we try to use the contrastive learnings with transcriptomic anchors to project this feature space into a unified one, and also on top of the biological observations. And for the last, we combine our framework with the downstream biological evaluations, so therefore it can provide a various analysis about our reasons. So here is the overview of our framework. So at the beginning, we have the input data. So as you can see, there shares 4 different omics layers, and it will fit into the omics-specific encoders. So they are parallel encoders. And in this part, we use the vector quantized variation autoencoders to learn the features independently from this input. So the reason is that we found that the variation autoencoders, it actually use the discrete distributions, which is more flexible, and also compared with other generative models, it sets no assumptions on the distributions of these data. So it is easier to approximate, like there may be different distribution for the different omics data. And for the next modules is the multi-omics contrastive learning. So in these modules, we use the contrastive learnings to make the transcriptomic anchors. Here, what we want to do is to use the other omics stores as the complementaries to the most important one, the transcriptomics. So firstly, we will construct the contrastive pairs between the transcripts and the other omics. And then we will try to summarize all these pairs together and optimize them simultaneously. And also, for the last modules, it's the biometric evaluations. So here we not only propose subtype identification models, what we want to do is also to provide the biological insights of our models to analyze results. So like here, we may have to analyze at the different levels, like we may have the single genes levels to analyze, or we treat them as the functional groups, or we can have more global ones like the pathway level analyze. And about the experiment statements, here in this works, we use the data set from 6 different cancer types. And for each of them, they consist of 4 different categories of the omics data. We have the genomes and the microRNA transcripts, the epigenomics, and the microRNA, the mRNA transcriptomics. As we can see here, for each data set, we have only a few sample sites, but for all of them, actually the feature size is huge. And the first result we want to show is the subtyping result. So this is the learned latent feature space by our models. And we can see that compared with the data distributions from the raw omics data, actually our model can make a clear distinction boundaries of all these clusterings and in the learned feature space. Like shown here, and we use the different colors to label the ground-truth cancer subtypes. And next shows the clustering performance. We shows the result for all of our 6 datasets. And we can see that for both of them, the post platforms can outperform the conventional method and also deep learning based method. For the conventional method, actually they use the traditional statistical models, and for the deep learning models, we have the different learning structures, like maybe the contents of the generator ones, and also like here we have the contrastive learning ones. And for the next result, we show the ablation studies. The first ablation studies is on the anchor omics. So here we show the result using the different omics anchors, and you can see that the Contrastive omics anchors can achieve the best performance. And also for the next, we show the ablation studies on the omics members. We try to decrease the omics sources used in these platforms, and also we can see that when we use full of these 4 omics resources, we can get the best performance. And for the next, we have the result of the bioevaluations. And here it shows examples of using the GSA, the gene set enrichment analyze. So what you want to do is to compare between the modules used with or without the biological observation mappings. So like here, the first one is we use these contrastive anchors, which is the transcriptomic omics, and compare with the no contrastive anchors ones. As you can see that actually when we use the transcriptomics as the contrastive anchors, we can get the subtypes with a more diverse functional lines. So therefore, we say that the MoCLIMs can independent cancel subtypes more reasonable because the resulting subtypes it actually has more distinct biological function characteristics. Okay. And for the next, we also evaluate our results in the pathway levels, and actually we can find that the differences found in the pathways may lead to the biomarkers or some potential therapy strategies. So like here we show some representative ones, the P43 signals and also the ERT signals. So I think this is all of my presentation today. And for the future work, we plan to conduct more in-lab experiments and evaluate the particle effectiveness of our models. And here you can find our papers.